now we're going to go on to talking about different types of microscopy. There's a lot of different types of microscopy, and there's a lot of different reasons we would use different types. So before we jump into the different types, there's some terms you need to know. Now, these two are very basic terms. Whenever you have a microscope, you need it to do two things for you. You need it to magnify the object. That means you need to make it look bigger. Okay, so magnification is a fancy word for make something look bigger. Okay, the other thing you need for your microscope to do is you need it to have resolving power or resolution. This is a fancy word to mean to show detail. So the example that I like here is you need to, if you were looking at your hand under a microscope, a bad microscope, you wouldn't be able to see all 5 of your fingers. But one with good resolution, you can see the detail. You can see each individual finger. So like when we're looking at bacteria under the microscope, our microscopes at home are not very good. So sometimes you're going to see like a whole bunch of stuff that's going to look like a blob, and you need to move over more to the edge of the slide where you'll see some good resolution where you can see the individual bacteria instead of just a big blob. So that's resolution. And you guys already know the parts of the microscope because you did a lab on this or you're working on it. So I hopefully don't have to go through all that. Okay, now a couple other terms. As you know, when you look under the microscope, I'll show you, there's 2 different lenses. The part you actually look through is the ocular lens. The ones we have at home are 10x, and we also have a 15x. Most of the time in the real world, the ocular is usually 10x. And then the other lens is the objective lens. And I think our microscopes at home have 3 different objective lens. They have a 4x, 10x, and a 40x. Whenever you want to calculate the total magnification, or TM, that's all you do is multiply the ocular times the objective. So if you're looking under your 40x objective and 10x ocular, your total magnification is 400. All right, and I think everybody got that one right on the lab questions. If you were looking just under the objective lens produces something called the real image. And when you're looking under both the ocular and the objective, that produces something called the virtual image. Now, let's go through the different types of microscopy. And the book is very detailed on this, but I'm just going to tell you the keys. I like to give the keys. The first type of microscope is a bright field microscope. These are the ones you will be working with at home. So bright field, just like it sounds. The background is going to be bright. So, or like, you know, light in color, bright. So the background is bright. The field is bright. The specimen, the bacteria that we're going to look at, will be darker because we're going to stain them. So as you can see here, the bacteria themselves, the specimen, is dark, or the specimen is darker than the background. And this is an actual picture of a specimen that I took at home with and the microscopes that we have. Now, when you give me your lab reports each week, don't just send me this, try to zoom in, and you know, this is the same picture. These two are the same picture, but I zoomed in my PowerPoint, and I cropped out a nice area so I could actually see what the bacteria look like. So try to do that so you could really see the shape and size of the bacteria. Okay, so that's bright. This is a bright field microscopy. The next type is similar, but it's dark field. So in dark field, the background is dark. You have a dark background, but you have a nice bright or light specimen. Now, this is a really specialized type of microscopy. It might be used if you want to look at live bacteria. In our lab at home, we will not be looking at living bacteria. We are going to kill our bacteria before we look at them. So we're not going to do this, but it would be used for looking for live specimens. The next type is called phase contrast microscopy. This is a very specialized type of microscopy. These types of microscopes are very expensive, okay, like tens of thousands of dollars. So obviously we don't have one of these at home. Well, if you have one of these at home, let me know. I'm coming over to your house. Okay, so these are very special specialized microscopes. Now, why you would do this is if you wanted to see intracellular structures. So if you wanted to look at a cell and you wanted to see the different organelles, if it was a eukaryotic cell, you want to look at the nucleus, you want to look at the endoplasmic reticulum, that's when you would use the phase contrast. The next type is a fluorescence microscope. So just like it sounds, fluorescent microscope, you're using some sort of fluorescent marker. So that's the first thing you need is some sort of fluorescent marker. So this could be that you use a special kind of fluorescent dye. There's some dyes out there that are fluorescent. There's many chemicals out there that are fluorescent. Or you could use an antibody that you put a fluorescent tag on. We're going to talk about that this semester as well. So you need some sort of fluorescent marker. You also need an ultraviolet light, a UV light, so that you can actually see the sample. Now, why would you ever use this? In medicine, we use this a lot for diagnosing infection. An example would be if somebody had some sort of lung infection. This is actually an image of lung tissue. I'm guessing that usually the green is the actual lung tissue itself. So that has a green fluorescent dye. And then this orange looks like it could be maybe a bacteria, like a Mycobacterium tuberculosis infection. So we would use this to try to look for bacteria in a tissue sample. That's one of the reasons we would use fluorescence microscopy. But again, this is kind of an expensive thing to do, so not something we would just do at home. This would be done in a professional lab. The next type of microscopy is scanning confocal microscopy. This type of microscopy is good if you want to look at different layers or multiple depths of a specimen. So like what this image is showing you is you can see the inside of the cell in this image, but then you can see the outside on this one. So you're seeing different layers of the same cell using this scanning confocal microscopy. Okay, the last type of microscopy is electron microscopy. So what's unique about electron microscopy is instead of using light, so instead of using light, all the other microscopes we talked about use light. These use a beam of electrons. And what makes this type of microscopy so special is it has tremendous resolving power. So you can see very, very fine details, super fine details. You can see every single organelle in the cell, crystal clear. You can even look at viruses. The only way to look at viruses, because viruses are so small, you need an electron microscope. So write that down. To look at a virus, you need an electron microscope because they're just so small. The other thing about electron microscopes is they can magnify between 5,000 to 1,000,000 times. So remember, the microscopes we have at home only magnify 400 times. That's nothing. Electron microscopes can magnify 1,000,000 times. So you can see the smallest thing in the world with these. There's two types of electron microscopes. The first are transmission electron microscopes, TEMs. With these images, you will get a two-dimensional image, but it'll be very detailed, so you can see the very small details within the virus or within the bacteria. The second type are scanning electron microscopes. So with scanning electron microscopes, you can get a 3D image. This technique uses a metal-coated specimen with electrons, and you scan back and forth over the specimen to get a really detailed image, 3D. Now, if you ever want to purchase an electron microscope, you can get a really good bargain. They cost about $1 million. So that's to tell you, that's the cheapest one I could find, $1 million to get an electron microscope. So these people who are studying viruses, this is not cheap, okay? You need these really special equipment. By the way, in the textbook, you'll see a lot of these really wonderful images that are taken by an electron microscope, but in real life, they are black and white. When you look at them under the microscope, they are colorless, but the color gets added in afterwards. So that's all manipulated. It's added by a computer. All right, so that's all of the different types of microscopes. And I made a table here. I think what you should do is fill out the table yourself. And then if you want me to check it over, send it to me in email and I'll check it over. Okay?